Welcome back to ICSE MasterClass. Today we're taking on the absolute core of biology, cell division. We're going to break this down in a complete explainer. So, if you've been looking for that perfect, structured guide to make your board preparation entirely stress-free, well, you're exactly in the right place. Grab your notes and let's dive right in. Listen, every single mark is worth your effort, and I'm going to show you exactly how to study smart. Today, we're figuring out how cells actually divide to help our bodies grow, heal injuries, and replace dead cells. Think of it kind of like a massive factory that has to duplicate its incredibly complex master instruction manual just to build an exact, fully functioning replica right next door. Getting a perfect score in biology is an absolute reality when you actually understand these core concepts rather than just blindly memorizing them.
All right, here's our syllabus for the day. We'll cover chromosomes and DNA basics, the cell cycle journey, phases of mitosis, animal versus plant cytokinesis, meiosis, and genetic variation, and we'll wrap up with our masterclass exam prep.
Let's jump straight into Section 1, Chromosomes and DNA Basics.
Okay, major board alert here. Examiners absolutely love to test if you can clearly differentiate between chromatin, chromosomes, and genes. So when a cell is just hanging out and not dividing, its genetic material looks like a long, thin, uncoiled, tangled thread. That's your chromatin. But the moment the cell prepares to divide, that chromatin tightly coils and condenses to form the distinct, thick structures we call chromosomes. And what about genes? Well, genes are literally just specific sequences of nucleotides sitting on that chromosome that encode particular proteins. They're the fundamental units of heredity that get passed down from parents to offspring.
Let's zoom in and break down the DNA itself. DNA is this massive macromolecule built of repeating units called nucleotides. Now, each nucleotide contains three distinct parts. You've got a phosphate group, a pentose sugar, and a nitrogenous base. These bases pair up perfectly across the two complementary strands like rungs on a ladder, held together by hydrogen bonds. And hey, remember this golden rule for your exams. Adenine always pairs with thymine, and guanine always pairs with cytosine. A with T, G with C. Never forget that.
But here's a crazy question. How on earth does 2 whole meters of DNA fit inside a microscopic cell? The answer is brilliant biological packaging. Think of 8 histone proteins acting together like a spool, and the DNA is a thread wrapping tightly around it to form a structure called a nucleosome. In fact, that chromatin material we talked about is actually made of about 40% DNA and 60% of these histone proteins. It really is an absolute marvel of engineering.
You are definitely going to need to draw this in your exam. When you're sketching it out, make sure you clearly label the exact attachment point in the middle, that's the centromere, and then label the 2 identical arms, which are your sister chromatids. When you look at a duplicated chromosome right at the start of cell division, it consists of these 2 sister chromatids joined right at that centromere. This exact spot is crucial because it's where the spindle fibers attach later on. Oh, and a quick reminder. Out of your 46 total chromosomes, 44 of them are autosomes that determine your general body traits, while those last 2 are your sex chromosomes, which determine gender.
Moving on to Section 2, the cell cycle journey.
The cell cycle is beautifully simple. It functions as this continuous, looping journey for the cell to divide, grow, and then redivide. It basically consists of 2 main parts, a non-dividing phase called interphase and the actual dividing phase called mitosis, or the M phase. Now, please make sure you don't fall into the common trap of calling interphase a resting phase. It is anything but resting. For most of its life, the cell is hanging out in interphase, actively and intensely preparing itself for the massive undertaking of dividing.
I want you to mark this heavily in your notes. Interphase has 3 distinct steps. First up is the G1 phase. This is the first growth phase where RNA and proteins are synthesized, but you won't see any visible change in the chromosomes just yet. Then comes the absolutely critical step, the synthesis, or S phase. This is where actual DNA replication occurs. The master instruction manual is completely copied. Finally, we hit the G2 phase, where the cell undergoes more growth and makes its final preparations for mitosis.
All right, Section 3, Phases of Mitosis. Let's get into the main event.
The highly choreographed dance of mitosis kicks off in prophase. This is where those duplicated chromosomes finally become distinct and visible, and the centrioles start moving apart to opposite poles of the cell. If you look closely, you'll notice the nuclear membrane and the nucleolus actually start to disappear. Spindle fibers begin to show up between the daughter centrioles, creating what we call the achromatic spindle. And pay special attention to the radiating microtubules, called asters, forming in the animal cell. You'll need to remember that specific detail for later.
Now for the next step. And guys, this is a frequently asked board question. In metaphase, all the chromosomes line up perfectly along the equatorial plane of the spindle. Each chromosome attaches to the spindle fibers right at its centromere. It forms this very distinct single-file line directly across the middle of the cell. An easy way to remember this? Think M for metaphase, M for middle.
Suddenly, boom, we hit anaphase. The centromeres split and those sister chromatids are rapidly pulled apart by the shortening spindle fibers. They are forcefully drawn toward the exact opposite poles of the cell. Notice how they bend into these little V shapes as they're literally dragged through the thick cytoplasm.
Let's look at Section 4, Animal versus Plant Cytokinesis.
As the nuclear membrane starts to reform in telophase, the chromosomes thin out and turn back into those tangled chromatin fibers. The two sets of daughter chromosomes have officially reached opposite poles. So, nuclear division is complete, but we're not quite done. The cell itself now has to physically separate its cytoplasm to become two distinct entities. This process is called cytokinesis. Let's see how this differs depending on the type of cell we're looking at.
You need to cement these visual differences into your brain. Animal cells are flexible, right? So they use a deepening cleavage furrow. It pinches in from the outside moving toward the center. Plant cells, on the other hand, have a tough, rigid cell wall. They can't pinch, so instead they must build a cell plate from the center outward to separate the two new cells. Furthermore, recall what we mentioned earlier. Animal cells have those star-like asters radiating from centrioles during division. Plant cells do not have centrosomes, so they don't form asters at all. Section 5, Meiosis and Genetic Variation.
Up until now, we've talked about mitosis. But now we introduce a completely special kind of division used exclusively for producing gametes, things like sperm and egg cells. This is meiosis. The absolute most crucial point here is that meiosis is a reductional division. Its entire purpose is to halve the chromosome number, so that later, upon fertilization, the normal diploid number, or 2n, is perfectly restored. So instead of creating identical clones like mitosis does, meiosis takes a diploid parent cell and creates haploid daughter cells that have only half the regular number of chromosomes.
And this brings us to one of the coolest parts of biology, crossing over. This is where chromatids actually exchange genetic material at a physical connection point called the chiasma. This process is exactly what creates the genetic variation that makes every single one of us completely unique. While maternal and paternal chromosomes are separating in the first division of meiosis, their non-sister chromatids form an X-shaped structure. They literally swap pieces of DNA. This massive mixing up of genes is the very reason you have your own unique set of traits. Finally, Section 6, MasterClass Exam Prep. Let's lock in those marks.
I want you to take a mental screenshot of this table right now. Comparing mitosis and meiosis is an absolute favorite for board examiners. Remember, mitosis occurs continuously in your somatic or body cells for growth and replacement. It maintains the full diploid set of identical chromosomes. Meiosis, on the other hand, occurs only in reproductive cells to form gametes. It results in haploid cells that are randomly assorted, driving all that amazing genetic variation.
Let's test what we've learned. Look closely at the simulated exam question. See how the chromosomes are lined up perfectly along the equator? So based on what we discussed, which phase of mitosis must this be? If you said metaphase, you nailed it. M for middle, right? Also, take a look at the poles. Notice the presence of those radiating asters? That tells us immediately, without a doubt, that we are looking at an animal cell, not a plant cell. Understanding how to spot these specific visual cues is your key to scoring maximum marks.
Make sure to join our ICSE MasterClass community using the links in the description below. We've got loads of exam prep material dropping across Instagram, YouTube, our website, and our Telegram channel. All right, before we wrap up this explainer, I'm going to leave you with a provocative thought to ponder. What exactly happens to our biology if the cell cycle somehow forgets how to stop dividing? Think about that for a second. How could the very mechanism that gives us life go fundamentally wrong? Keep studying smart, keep asking questions, and I'll see you all in the next explainer.